Sharks don't get cancer. If that statement were true, it would be an extraordinary discovery in the medical field. After all, almost all animals, including humans, are susceptible to this disease. So what's the secret behind sharks' apparent immunity to cancer? First of all, let's make it clear that sharks are not 100% immune to cancer. There have been cases of cancer found in various shark species, but it is true that they seem to have a much lower rate of developing the disease compared to other animals. For example, a 2006 study found no cancer in any of the 319 sharks that were examined. Even more impressive is that these sharks ranged in age from 4 to 70 years old. Considering that cancer is largely a disease of older individuals, the researchers felt that if cancer were a common problem for sharks, they would have found some cases in the 70-year-old sharks. Since then, there has been one reported case of cancer in a shark, which makes sense considering that sharks have been studied far more extensively since then. But what about all the other animals that get cancer? Why don't sharks seem to be affected by this disease like every other creature on the planet? In order to answer that question, scientists have turned to shark cartilage. Shark cartilage contains a lot of cartilage cells, which are the cells responsible for building and maintaining the structural integrity of the skeleton. One of the hallmarks of cancer is the uncontrolled growth of cells, and that includes cartilage cells. So scientists have been studying how shark cartilage cells regulate their own growth in order to learn how to prevent cancer from occurring in other animals. One of the things they've discovered is that shark cartilage cells have a very low rate of mutation. In order for cancer to develop, cells need to acquire mutations that allow them to grow and divide uncontrollably. The fewer mutations a cell has, the less likely it is to develop into a cancer cell. And shark cartilage cells seem to have very few mutations, even when compared to the cartilage cells of other animals that do and get cancer as often as sharks do. Another thing scientists have discovered is that shark cartilage cells have a high rate of apoptosis. Apoptosis is a form of programmed cell death that occurs when cells become damaged or begin to grow out of control. It's like a built-in failsafe that prevents damaged cells from becoming cancerous. In other words, if a shark's cartilage cells start to mutate and grow out of control, they will die before they can turn into cancer cells. So it seems that sharks' resistance to cancer comes down to two factors: low mutation rates and high apoptosis rates. But why do sharks have such low mutation rates? And why do their cells undergo apoptosis when they start to grow out of control? Scientists are still working to answer these questions, but they have some clues. One possibility is that sharks' natural defenses against predators may also protect them against cancer. Sharks have very efficient immune systems that are constantly on the lookout for threats. This may help to eliminate any cells that start to grow out of control before they can become cancerous. Additionally, sharks live in a salty environment, which may create an osmotic stress that helps to keep their cells healthy and functioning properly. The bottom line is that we do not know for sure why sharks have such low mutation rates and high levels of apoptosis. But it's clear that these two factors play a major role in their apparent immunity to cancer. Ultimately, understanding how sharks resist cancer could help us develop new treatments for cancer in humans. By studying these amazing creatures, we may be able to find ways to prevent the disease or even reverse it in its early stages. So next time you're at the beach and see a shark, remember that it may be more than just a scary predator. It could be the key to unlocking one of the greatest mysteries of our time. If you enjoyed this video, please leave a like and subscribe for more science videos. Thanks for watching.